The axolotl, a fascinating amphibian native to Mexico, possesses an extraordinary ability that seems almost like biological time reversal, the capability to regenerate entire limbs, spinal cord, heart tissue, and even parts of its brain. But how does this work, and why is it important? Unlike humans, whose wounds heal by forming scar tissue, axolotls can fully restore lost body parts, making them a key model for regenerative medicine and the study of cellular time perception. Imagine if a human could regrow an amputated limb or recover from spinal injuries without complications. This is precisely what scientists hope to understand by studying axolotls. Their unique regenerative process involves the formation of a structure called the blastema, a cluster of undifferentiated cells that revert to a more primitive state, essentially rewinding their biological clock to regrow tissues. Could this mean that time, at least at the cellular level, can be manipulated? If so, what implications might this have for anti-aging therapies and the reversal of degenerative diseases? Researchers have already identified genes in axolotls linked to regeneration that are also present in humans but are inactive. So, what if we could switch them back on? Experiments have shown that introducing axolotl regenerative factors into mammalian cells can enhance wound healing, hinting at future breakthroughs in regenerative medicine. For instance, in cases of severe burns, organ damage, or nerve injuries, activating a regenerative response similar to the axolotls could transform medical treatments. Additionally, their ability to repair brain tissue raises intriguing questions about memory and consciousness. Does a regenerating brain preserve past experiences, or does it reset, effectively erasing its own history? Could this concept influence how we understand time at the neurological level? From extending human lifespan to revolutionizing treatments for amputees and spinal cord injuries, the study of axolotls challenges our perception of biological limits. What other species might hold hidden clues about reversing cellular aging? And could humans ever harness such abilities? Axolotls, Blastema mexicanum, possess an extraordinary ability to regenerate entire limbs, spinal cords, heart tissue, and even parts of their brain, a capability rare among vertebrates. Unlike mammals, whose tissue repair relies on scarring, axolotls reactivate embryonic-like cell programs, forming a a mass of undifferentiated cells capable of recreating complex structures. This process involves molecular signaling pathways such as the Wnt, BMP, and FGF pathways, which coordinate cellular dedifferentiation, proliferation, and patterning to regrow missing body parts. If an axolotl loses a limb, within days a blastema forms at the wound site, and over a few weeks, it regrows a fully functional and anatomically identical limb with muscles, bones, nerves, and blood vessels. This regeneration is like a biological time machine, resetting damaged tissue to an earlier developmental state, effectively rewinding the clock on cellular damage. Axolotls exhibit a form of biological time reversal at the cellular level. When injured, their cells revert to a pluripotent-like state, mimicking early embryonic development. Unlike human stem cells, which require external reprogramming to become induced pluripotent stem cells, iPSCs, axolotl cells naturally reset themselves to an earlier state, enabling tissue reconstruction. This unique ability challenges traditional notions of cellular aging and suggests that biological systems might possess intrinsic mechanisms to rewind developmental time under specific conditions. Researchers have observed that when axolotl limb cells undergo regeneration, they lose their mature characteristics temporarily, resembling embryonic progenitor cells, before differentiating into new limb tissues. This process is like erasing and rewriting a saved computer file, removing old instructions and replacing them with new ones. to restore function seamlessly. Unlike humans, whose genetic and epigenetic markers prevent extensive regeneration, axolotls regulate gene expression in ways that maintain regenerative potential throughout life. Epigenetic modifications such as DNA methylation and histone modification allow axolotl cells to remain flexible and responsive to regenerative signals. Additionally, axolotls express specific genes such as FTC1, LIN28, and MIZX2 that enhance regenerative capability while suppressing fibrosis genes that cause scarring in mammals. A study on axolotls identified that their ability to regenerate spinal cord tissue is linked to the suppression of the p53 tumor suppressor gene, allowing cells to proliferate without forming scar tissue. Their genetic regulation works like a well-maintained software system, keeping old functions available for activation when necessary, unlike human biology, which tends to lock past developmental capabilities. Understanding axolotl regeneration could revolutionize regenerative medicine, particularly in healing spinal cord injuries, treating neurodegenerative diseases, and regenerating lost or damaged organs. By deciphering how axolotls activate regenerative pathways, scientists hope to induce similar mechanisms in humans. Experimental approaches using gene editing, CRISPR, and stem cell technology aim to replicate axolotl-like regeneration by reactivating dormant regenerative genes in mammals. Researchers have successfully activated partial regenerative pathways in mice by introducing axolotl genome, showing promise for spinal cord repair therapies in humans. Applying axolotl-like regeneration to humans is like unlocking a hidden function in a smartphone, an ability evolution suppressed but might still be reactivated with the right programming. Bioelectric signaling, the regulation of cellular processes through electric potentials, plays a crucial role in axolotl regeneration. Research shows that axolotls use ion channels and voltage gradients to signal cell growth and tissue patterning. By manipulating bioelectric fields, scientists have induced regeneration in non-generative species, suggesting that electrical cues act as a fundamental mechanism guiding tissue repair. Experiments in frog tadpoles demonstrated that altering bioelectric gradients can trigger limb regeneration, a trait normally absent in adult frogs. Bioelectric signals act like a GPS system, guiding cells toward their correct positions to rebuild complex structures after injury. Axolotls exhibit negligible senescence, meaning they do not show typical signs of aging like reduced metabolic efficiency or increased disease susceptibility. Their regenerative capabilities may contribute to their longevity by continuously replacing damaged cells and tissues. This feature contrasts sharply with mammals, where regenerative decline is a hallmark of aging. Understanding how axolotls evade cellular aging could offer insights into extending human health span. Unlike mammals, axolotls maintain telomere length throughout their lives, preventing the chromosomal deterioration associated with aging in humans. Axolotls are like self-repairing cars, constantly replacing old or damaged parts, ensuring they function optimally for an extended lifespan. One of the most astonishing aspects of axolotl biology is their ability to regenerate brain tissue without losing cognitive function. Studies show that axolotls can regrow large portions of their brains, yet retain previously learned behaviors. This suggests that neural networks can be reconstructed in a way that preserves functional memory, a phenomenon with profound implications for brain injury recovery in humans. Experiments where axolotl brains were partially removed demonstrated full regeneration within weeks, with the animals retaining their learned responses to stimuli. Their brain regeneration is like rebuilding a damaged hard drive while keeping all stored data intact. Axolotl regeneration raises intriguing questions about how biological systems perceive and manipulate time at the cellular level. If cells can revert to an earlier developmental state, does this suggest that biological time is flexible rather than strictly linear? Regenerative processes in axolotls might indicate a form of biological time travel, where cells revisit previous developmental states to rebuild lost structures. Some researchers propose that studying axolotl regeneration could redefine our understanding of aging, development, and cellular memory, potentially challenging traditional models of biological time progression. This concept is similar to rewinding a movie to a specific scene and playing it again with perfect resolution, rather than simply pausing and resuming from where it left off. While axolotl research presents groundbreaking potential for human medicine, there are ethical considerations in applying these findings to humans. Genetic manipulation to enhance human regenerative abilities raises bioethical questions about unintended consequences, such as uncontrolled cell proliferation or immune rejection. Future studies aim to balance regenerative advancements with safety considerations, exploring alternative approaches such as biomimetic scaffolds and tissue engineering rather than direct genetic modification. In trials attempting to induce limb regeneration in mammals, scientists have faced challenges in preventing excessive or malformed tissue growth, highlighting the need for precise control over regenerative processes. Applying axolotl-like regeneration to humans is like hacking a system. You need to ensure stability and security before enabling new functions.